Little disclosure, obviously I have an accent because I wasn't born here. I am an immigrant. They let me in. Now I vote, which I did this morning. My accent is Swiss-German. I'm from the city of Basel in the north of Switzerland, where Germany, France, and Switzerland touch. And I have a fairly good excuse for my accent given today's topic, and that is the gentleman who discovered DNA. It wasn't Watson and Crick. They discovered the structure of DNA. But the guy who actually discovered DNA was a physician called Friedrich Mirscher, and he was from my hometown. And had he spoken English, he would have had a similar accent to me. He discovered it in the pus of patients that he was treating, in the bandages, and later made the best preparation from salmon sperm from the Rhine River. So today's topic is changes that made us human, and you probably expect genetic changes. I will talk about genetic changes, but my big take-home message is that what made us human is a package of genetics and environment. So if we take a feat of recent human technology, which are the Landsat satellites, and step way back and use these satellite images of night over Earth, you can tell where humans live. That's where all the bright lights are. At least that's where the rich humans live because they have enough money to keep the grid on. There's plenty of humans who live in the darker spaces, but the high density of industrialized humans, you can see Japan, you can see all of the west coast of China, look at the density in Europe, North Africa, you can see the Nile River lit up. In Africa, you can see South Africa, you can see Brazil, Argentina, and then look at the US, how much denser the East Coast is than the West Coast. And those colors indicate where the great apes live. The great apes are our closest living relatives. There's several species alive. And we know our relationships because we now can sequence the entire genome. And that tells us that two of these great apes, bonobos and chimpanzees, are more closely related to us than they are to any of the other great apes. So we are an ape. The word great ape doesn't really make sense because that was classical humans must be special, and the great apes are these other things that are a bit more furry than we are. But actually there is chimpanzees and humans, and then all these other apes. But you can call them all hominids. That also works. So based on genetic comparisons that started in the 80s, where people would read 100 letters of DNA and compare those, there were big arguments, who is more closely related, gorillas or chimpanzees? We now know that humans and both species of chimpanzees share the common ancestor between 6 and 7 or 7.5 million years ago. The precise timing is still unclear. Why do we know this? We have access to the entire genome. So every single cell, I'm here to remind you that you started out as a fertilized egg, and we're going to make a model of an egg. That's an ostrich egg from San Pasqual Valley. I think the ostrich farm is not there anymore, but a few years ago I bought it. So if you make a human egg, which is the size of a grain of salt, you make it 1,000 times bigger, the sperm would be about this big. So the reason you sit here today is that at one point, one of the hundreds of millions of sperm that your father generated met an egg that your mother generated. And they formed a new diploid organism. So at this scale, a genome is actually ridiculously big. The genome in each of your cells at actual size is about 1 yard long. So 1,000 yards is this big, but that's only half the genome. So in every one of your cells, you have 2 times 3 billion letters. And at a scale of 1 to 1,000, that's how big your genome is. And in early genetics, we would be really happy if you could sequence a couple of hundred letters. And over the course of 20 years, we now can sequence the entire genome of multiple apes and thousands of humans. Not a week goes by without a fantastic new piece on the settlement of Australia, for example, or another extinct hominid, a human that's 20,000 years old somewhere in Romania, that they managed to sequence ancient DNA. So the genome, indicated here by a model 1,000 times bigger than it actually is a treasure trove of information that allows us to learn a lot about past evolution. Now, you've probably all heard, oh, you're just a little bit over 1% different from a chimpanzee. That's actually wrong. If you take the entire genome that we can, that's the difference between you and a chimpanzee. That's about 5% of this 1 kilometer. And that's surprising somehow. It's 5%, it's more than 1%, but it's not that much. If you take your DNA, line it up with that of a chimpanzee, then all the differences between you and the chimpanzee will be about 5%. Why did it jump from 1% to 5%? In the old days, it was very difficult to sequence through pieces of DNA that are highly repetitive, because it's like a hall of mirrors with a sequence going this way, and then this way, and then this way again. Now, scientists have figured ways to sequence that, and they have found that the reason it is 5% and not 1% is that humans have huge chunks of DNA that you don't find in a chimp, and chimps have chunks of DNA that are private to them that don't exist in humans. So if you include the pieces that are missing from humans and chimpanzees and the differences, you get to 5%. So I just thought I'd put in context how vast a genome is, because you hear these, oh, you can have your genome sequenced and then figure out what your ancestry is. It's extremely complicated. How does a cell like that manage to package this DNA into its nucleus? It's a very complex question. It has to do with the fact that the cell uses something like spools. It spools the DNA around proteins. These proteins are called histones. And this gives you the scheme of how you get from a strand of DNA that you're all familiar with to a chromosome that you can observe when a cell is actively dividing. When you look at a chromosome, which for many years was the only DNA scientists could see, they had to catch a cell in the act of dividing, splatter that cell on a glass slide, and then if they were lucky, they would find these beautiful double chromosomes that are in the process of dividing. That's called cytogenetics. And counting up the chromosomes in a cell is called the karyotype. And for a long time, that's the closest scientists got to DNA, because they were not capable of doing next-generation sequencing, where they can sequence the entire 3 billion letters times 2. Every one of these cells is diploid, right? There's 2 copies, one from dad, one from mom. Each copy is 3 billion letters. And we now have the capacity to read through those. But when a cell is busy doing its work, and being a liver cell or an eye cell or a cell in your pinky, most of the DNA is tightly wrapped around spools called histones, except the areas that allow that cell to do its work as a muscle cell or an eye cell or a liver cell. So this is blowing up and going into one of these string of beads. You see that a bunch of proteins in purple, yellow, and green are the proteins that allow you to compact your DNA as tightly so that it fits into the nucleus. And below is an electron microscopy image of the beads on a string pattern of chromatin, a mixture of DNA and protein. It turns out that this packaging contains information. People usually think simplistically, it's in your DNA. Yes, it's in your DNA. That's also your DNA. If an area of your DNA is tightly packaged, that area is not going to be used by a particular cell. So cells can regulate which part of the DNA is playing by either packaging it very tightly or opening it up so that the gene reading machinery has access to it. And so let's go back in the days, over 20 years ago, 1982, people noticed, well, if you take these chromosomes and you stain it with a chemical called Giemsa, that chemical is going to stain the part of DNA that has a lot of AT letters as opposed to GC. And then you get these dark bands. What you see here is chromosome 1 to 23 of human, chimpanzee, gorilla, and orangutan lined up next to each other. Can you notice how similar they look? The banding pattern is very similar. Where you have dark bands, there are almost no genes. Where you have light bands, it's GC rich, that's where most of your genes are sprinkled throughout it. So many years ago, people had noticed, hey, it's amazing. Chromosomes of humans and their closest living relatives look very, very similar. There's one big difference. The second chromosome, you can see that the apes have 2 chromosomes and we have 1. That's because sometimes in the last 5 million years, we took 2 ape chromosomes in our genome and fused them together to make our chromosome 2. That's why humans have 23 chromosome pairs and apes have 24. But it turns out the 2 ape chromosomes, if you put them together, they still look very similar to our fused chromosome. They're not all apes. These are lesser apes, the gibbons of Southeast Asia. They also are tailless primates with super long arms. They're famous for brachiating. Anybody here has been to the San Diego Zoo and seen the gibbons? About a third of people. I highly recommend it. If you go there over lunch, they tend to vocalize. They're totally monogamous, sometimes in series, but it's a couple that controls an area, and they duet. They go woo, 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 like that, and they compete against other monogamous pairs. Well, it turns out that their chromosomes have hit the fan. Something happened in their genome. We now know what, based on a paper that came out 2 years ago. A little piece of DNA jumped around the entire genome from chromosome to chromosome, and after it went everywhere, it caused these chromosomes to break and mix. So their chromosomes look completely different than the chromosomes of the ape. You can get their chromosomes, sort them out, put a fluorescent tag on them, and then take those chromosomes one at a time and throw it against chromosomes of a human. And you can see how different gibbon chromosomes light up the human chromosomes in different colors. And I show you this image because this is the contrast between the chromosomes in a dividing cell, those little sticks, those little cigars, and the way chromosomes look in a cell that is busy doing its stuff. It's not dividing. So that nucleus that looks like a moon with all these different colors, this fuzz, those are the chromosomes that are puffed up so that the proteins that are reading the DNA and transcribing it into RNA to make proteins can have access to it. So your genome alternates between being in tight little packages and being a kind of fuzz that allows your cell to go and read a particular string on. And it turns out if you take these gibbon chromosomes and you throw them on the chromosomes of humans and the great apes, chimpanzees, gorillas, orangutans, and even macaques, you can see the last one to the left looks quite a bit different. Macaques are distantly related to us. They've changed their chromosome quite a bit. But the first four, the so-called great apes and us, overall, the color scheme looks very, very similar. So this is the powerful technique known as chromosome painting or cross-species painting of chromosome with fluorescent probes, and it allows you to study how these chromosomes have changed over time. And in a nutshell, the chromosomes haven't changed that much between us and the great apes, except for our second chromosome being a fusion of these two chromosomes in the great apes. Now, you can take a phylogeny, you can ask, where have which changes of chromosomes happened? Chromosomes can either fuse, as I already mentioned, or they can have an inversion where a piece of DNA that's on one arm jumps to the other arm. They can break. And if you plot these out, it turns out you can just use the chromosomes. You haven't sequenced any DNA. And you can retrieve that, in fact, humans are more closely related to the two species of chimpanzees than they are to gorillas or to orangutans. The problem with how chromosomes change is we have no, it's not a mutation rate, it's not a clock. We have no idea what determines how quickly these chromosomes change. But they're very powerful to tell us that, again, this is not DNA sequence, this is how the DNA is packaged, puts us and the two species of great apes known as chimpanzees and bonobos as most closest living relatives. So what is counterintuitive is, you think, oh, I'm from this lineage, I descend, or I'm European, so I must have some ancestors that come from here and there. The complicated thing is your DNA doesn't come in a package. Each one of us in this room is a mosaic of little pieces of DNA, and each one of these pieces has its own history. Why is that? Because if you sequence the DNA of two individuals, think of it as a river flowing backwards. The river consists of males and females who breed. They have offspring. And the DNA can only pass up a generation if a male and a female meet and successfully reproduce. And that child survives until the child itself is sexually mature. Well, one piece of DNA that you find in two humans now that live in two populations could have a common ancestor in that particular individual and another piece in that other individual. And we all like that. We have chunks of DNA that we share with somebody else, and another chunk of DNA that we share with somebody completely different. So next time you meet somebody who talks about pure bloodlines or, purebred, it's all complete BS. There is nothing purebred. There is only mosaics of things that are mixed. You can inbreed things like we do in the lab with our lab mice, We cross siblings for 9 generations. Now all the mice are like a clone. That's purebred, but that is known not to do too well in the wild. In the lab, it's fine, because there's no pathogens, no predators, and we like to have an isogenetic backdrop to our mice. But it's totally artificial. In the wild, when populations do that, they become inbred and they tend to die out. So the fact that each of us is a mosaic is very important for our survival. So I thought I'd illustrate this with a little cartoon. This is you with a Y chromosome, a mitochondrial DNA, which happens to be a little circle of only 16,000 letters, and then your chromosomes. Autosomes, which are the chromosomes that have nothing to do with sex, with male or femaleness, and then sex chromosome, the Y and the X chromosome. Now you have 2 parents, and your parents each have 2 parents, and they have twice as many great-grandparents. So what's really interesting is that every generation as you go back, you have 2 times the number of ancestors. My rule of thumb of sociology is usually with about 10 people, you have 1 person that's not so nice or an idiot. And so the bad news is, as you go back to your ancestry, these groups get really big, so we probably all have fairly big idiots in our ancestors. The good news is you don't inherit everything from all these people, because as they reproduce, there is a random part, you only get half of each parent. But they are all shuffled. And so you are a mixture of a complete mosaic in most your DNA. The only 2 pieces that are not mosaic are mitochondrial DNA that you inherited from your mom. If you're a guy, you will not pass it on. If you're a woman, you will pass this on, just like a family name is in the patriarchal societies. And so the Y chromosome behaves like a family name and is very powerful to study migration and colonization and admixture. So before I continue with genes, I try to convince you that we are much more than a bubble that contains DNA. So what are apes, and I'm including us, made of here? You all know that people talk about DNA or the genome as the blueprint of life. It's the molecule of inheritance. That's the key molecule passed on through generations that determines that you grow up to be human and not chimpanzee or macaque. And it is a template. It is transcribed into messenger RNA, which is then translated by a machinery of RNA and proteins, which makes proteins, like you have in egg white. But that's not you. A cell always contains other things, namely sugars, and it contains fats. So just when you want to bake a good cake, you need carbs and you need fat if you want a good cake. There might be alternatives these days with vegan and, but let's talk about classical cakes. So every living cell is really a bubble, the nucleus, that contains the DNA, surrounded by an ocean of cytoplasm that has a lot of other small bubbles like the Golgi and the lysosome. And then around the whole thing is another fat bubble, the membrane. And on the outside of that fat bubble, it's not smooth, it's completely packed with sugars. And these sugars, it turns out, play a lot of roles in telling other cells what this cell is about. When this sperm got to the egg, the only reason this sperm got to the egg is that the female didn't kill it. The other 200 million sperm were all killed by the female, but this one made it to the egg. And what is the sugars on the outside of the sperm had a lot to do with the fact that it met the egg and managed to make you and I. So the dogma of molecular biology that everyone is familiar with these days is that if you have a stretch of DNA, you can put it into bacterium, and then you want to make insulin, you want to make human insulin, just take the sequence for human insulin, put that in a bacterium, and then the bacterium will produce insulin. It's great. You don't have to use pig insulin. You can treat a lot of people. But that works good with insulin because insulin doesn't have any sugars attached to it or fats. It turns out to make a cell, you need these lipids and these glycans. And then the cell, all of us are billions of cells, these cells talk to each other. They have to decide when to make a muscle, when to make cartilage and bones, and the ectoderm, the mesoderm, and so forth, the different layers of the organism. And these sugars that we call glycans on the outside of cells, depicted in blue here, together with the lipids, you have no genes for sugars and lipids. You only have genes for proteins that can change sugars and lipids from one sugar to the other, but the sugar itself you have to eat, and the lipid you have to eat. So by definition, what you end up being as an organism is massively influenced by where you live and what you eat, what you consume. All these molecules talk to each other during development, and they don't do so in a vacuum. This dividing organism that goes from a fertilized egg to billions of cells that becomes you starts out in a womb, if you're a mammal, and then is born and is still developing, especially its brain. And this all happened under attack of microbes and parasites, under a physical environment that can be very hot and sunny, or it can be very cold and not much light. And very importantly for us, and some of the great apes, a cultural environment. You're not alone. There are others like you, older ones, and they might talk to you, they might teach you things, they might demonstrate things, you might want to copy them. And if you want to continue that process of passing on DNA, you have to find an opposite sex individual and convince her or him to have sex with you, and then you maybe can pass on the DNA to the next generation. So what is mind-blowing about hominids, but especially the lineage of hominins leading to us, as pointed out by a famous anthropologist, Loring Brace, is that we have come up with a non-biological inheritance system, which we call culture. It's something that we do, that we transmit to the next generation, and they also do it. It can change quite rapidly. It's different than DNA. Culture is really good at borrowing things. Right now you have the entire world that is mimicking Hollywood. There's no DNA for Hollywood behavior, but Hollywood is superbly influential around the world. People in China had to invent a word to say cool, because it sounds like cool, and it's cool. So now they say cool. The entire world says click. It's the most common word apparently used in all languages around the world, because we all click on computers these days. So culture is very rapidly evolving. It's transmitted in all directions. Little kids can teach their grandpa how to use an iPhone and watch a YouTube on some funny thing happening there. And human biology is completely perfused with culture. It has famously been said that there is no way of understanding human evolution without the context of culture. So just the very famous saying that biology makes no sense without the context of evolution, human evolution makes no sense without the context of culture, because we're so profoundly cultural. And it turns out that many years ago, many ethnographers around the world have gone around and have commented on the fact that every society around the world has kind of a basic toolkit, which is what allows it to create a niche. Niche is just the French word for a nest. It creates a surrounding that carries you as a species, and has been incredibly influential for the fact that we now colonize the entire planet, are in the process of changing its weather, and leading about half the species of large animals to extinction. Things like shelter, control of fire, tool making, weapons, twining or weaving, using containers such as this ostrich eggs to drink, cooking, the use of fire, levers, tying material, are found in every society that people have encountered around the world. Very importantly, we have this socio-cultural niche. We share symbols. We have personal names. We do not know of any other species of animal where individuals can gossip about others because they don't have names. Without names, how can you gossip? There is no way of saying, do you believe that chimp yesterday did this, if you don't have a name for that chimp. Tellingly, anthropologists like myself, when I studied chimpanzees in West Africa, we give the chimps names. One is called Rousseau, the other one Ulysse, or the one Frodo, or Fifi, and so forth. But the chimps do not know these names. They don't answer to them. We impose those on them because we just, we work when we talk about individuals. We want names. Very importantly, with the advent of more monogamy, just relatively more monogamy than in the great apes, kinship terms become very important. Great apes tend to not know who their father is, with the exception of gorillas, because usually there is one silverback that dominates reproduction for a long period of time. In chimpanzees or bonobos, none of the individuals have a way to guess who their father is, because the mother will mate with dozens of males for each egg she ovulates. In human societies, usually we have kinship terms. You know about your paternal uncle and your grandma and your auntie, and that allows you to build tribes, which you can't without names and kinship rules. We share rituals, dance, and music. We have sacred spaces. We have group identity. which has become incredibly important in competition between groups and cooperation between groups. The darker side is war between groups, which is very old. All these are phenomena that there are possibly inklings for in some of the really big brain species, including orcas, bottlenose dolphins, some pilot whales, maybe elephants. But the great apes don't show any evidence for these things. So as a summary of kind of modern human behavior, I really like this slide, which shows you 1 day worth of intercontinental flight. That's what we're pulling off. That's the ape we are. We have built flying machines on every given day. I was just in Europe for 10 days. You just get onto a metal tube with another 300 people you don't know, and you sit there patiently for 12 hours, and then you get out of that tube when you get on another continent. Sarah Hrdy, who's a famous anthropologist, physical anthropologist, is very interested in child rearing and the idea of cooperative breeding. She's now emerita at UC Davis, came up with this fantastic thought experiment. She said, what if you took 300 unrelated chimpanzee males who in the wild jealously guard their territory? If as a male you moved from your territory where you were born to the next territory, you die. What if you took these guys and filled a plane with them? How many do you think would still be alive after a flight from LA to Zurich? Probably not many. So as bad as we can be, as bullying as we can be and selfish as we can be, something has changed in our social and aggressive behavior where you can take 300 unrelated male humans, put them in a metal tube and fly them to another continent. What is very exciting these days about studying genomes, and I hinted at this before, is that some incredibly skilled genome scientists around the world, but the 2 hubs are at the Leipzig Institute for Evolutionary Anthropology, people around Svante Pääbo, and then also in Denmark, Eske Willerslev. They have been able to get DNA in tiny little tidbits from extinct species. I show you a list there of images of extinct megavertebrates that have been completely sequenced. These animals are extinct, like the quagga or the woolly mammoth, but we now have their entire genome. And the same is true for several hominids. And based on this, we know that most of us sitting in this room have tiny bits of DNA from a species that doesn't exist anymore, called Neanderthals. Fairly few in this room probably would have other pieces from a group known as Denisovans, which were the sister taxa of the Neanderthals. And there is some very interesting evidence that among African population, there is also a little contribution from people who are not around anymore, but used to live on the African continent. So there has been admixture from recently extinct species over the last 50,000 years. But what we know, the big pattern of how humans colonized that entire planet is that in the last 70,000 years, modern humans who clearly evolved in Africa, where they came up with projectile weapons, with the kinship networks, with the modern language, with that killer app, a kind of explosive combination of traits, cultural and genetic, spread through the entire world and bumped into some archaic humans, the Neanderthals in Asia and Europe, and then into some others in Asia that included the Denisovans and possibly Homo erectus all the way down in Flores. So what genome scientists can do now is they can take the genome of many humans and they can ask, where do we find little pieces of Neanderthal and Denisovan? And the answer is mostly in areas known as gene deserts. Those black stripes that were stained on the chromosomes where there are almost no genes, those are the only areas where genes that came in from a distant relative, the Neanderthal, with whom we had a common ancestor 500,000 years ago, 500,000 years ago, that's the only place where we tolerate that different DNA. Because it seems to interfere with how we function if it's near functioning genes. And that's a very strong evidence that, yes, we could mate with Neanderthals, we could have live offsprings, but these offsprings mostly paid a price. And our genomes purged the Neanderthal and the Denisovan DNA from all the areas that we know are involved in important function, including areas that have genes that affect the development of speech and locomotor control. But there's a few exceptions that prove the trend, that buck the trend. there's a couple of genes that have made it into modern humans that actually have been taken on by modern humans. The most famous one is the gene EPAS1, which is a gene found only in Sherpas and Tibetans, and it allows Tibetan mothers to have normal birth weight babies at 5,000 meters, that's 16,000 feet above sea level. Han Chinese, who now make the majority of people who live in the city of Lhasa, the capital of Tibet, when they get pregnant, they try to get down to Chengdu at lower elevation, or they will have a low birth baby. So there are a few cases where these archaic genes were borrowed and stayed in the genome, but they are the exceptions that prove the rule. So one of the big questions is, given that there were these other species around, how come we are the last one standing? You know, there was a little bit of movement of DNA between those, but somehow these anatomically and behavioral modern humans blasted out of Africa, took over the world, and did, you know, the jury is out what caused the extinction of the other ones, but quite a few people lean towards a certain role of us in the demise of Neanderthal and Denisovans. So before I dwell a bit more on what genes might be doing, I thought I'd refresh your genetic vocabulary. I already used the word genome, which refers to the totality of the DNA you have in every cell. 2 times 3 billion letters distributed on 23 chunks and 23 chunks. Now, if you go on the genome on a particular place, scientists tend to call that a gene or a locus. The old definition of a gene is a piece of DNA that encodes a particular protein. And you might be surprised to know that only 2% of your entire DNA makes up the so-called classical genes. So out of all this, only this encodes classical genes that we know have a promoter and then exons, and you can translate them, and you know it makes hemoglobin, or it makes keratin, or your other favorite protein. If you compare genomes from mice to humans, you will find there's another 2% that seems to not be allowed to change, because it's very, very conserved between mouse and humans. But it's not genes. It's just DNA sequence, but it doesn't translate into proteins. So people think this is really important regulatory DNA. So it's daunting, you know, for the 3 billion letters, you have this much protein coding and this much very strongly conserved regulatory DNA. What does all the other stuff do? Well, we don't know. People used to call it junk DNA. They thought it was just the stuff on your laptop, you know, when you write your essay and you do another version of it. Sooner than later, your whole laptop is completely full of stuff, all your extra copies. Maybe something like that is going on in the genome. Any place is called a locus, and so on the same chromosome, the loci can have numbers. It's just the Latin word for a spot. But any particular place on your chromosome, because you have 2 copies, can exist in 2 different, slightly different versions, and those are called alleles. So one allele from your father, the other from the mother. And comparing these can be very interesting. In fact, if one of them is broken because there was a mutation, the reason why you're alive and not dead is that the other one is not broken. It's a great advantage to have a mother and a father who are not closely related for this reason. If you're very inbred, they're both likely to be the same. And so you have more genetic issues. A haplotype, haplo is Greek for simple, is simply like a chain of beads where you have a couple of alleles that travel together in time. That's called a haplotype, and you can define those. I use little colors for slightly different DNA sequences. And so a particular locus, if it's a gene, it has a promoter that tells the machinery, come here. That's where I start reading and making messenger RNA here. The messenger RNA can be edited and spliced and then capped, and it's then sent to the ribosome, and you make protein. So that's kind of classical, going from a DNA instruction to the myoglobin that is in all of your red blood cells and allows you to carry gas around. Now, I find it relatively abstract to imagine 20,000 things. 20,000 is the number of genes in your genome, and you have 2 times 20,000 on each one of your copies. There's an artist called Spencer Tunick who he likes to make flash mobs of naked people. I was decent, so this is very far away. This is on the Grand Plaza in Mexico City. That's 20,000 people standing there naked. So these would be your key players in your genome. Each one of these persons is one of the genes. And imagine, do you think really that any brain of any scientist can have a concept of how these 20,000 players dance in every one of your cell types? Because the way they dance is different, whether you're a liver cell or a neuron. It's incredibly complex, but it doesn't stop there. It gets worse. So 20,000 to 22,000, that's the number of undergrads we have at UCSD. Now, there is thousands of pieces of DNA that get transcribed into RNA but never become a protein. Those are called long non-coding RNA genes. And there is hundreds of thousands of enhancers. Enhancers, if there is a gene somewhere here, the enhancer can be somewhere on another chromosome. But it's called enhancer because it has an influence on whether this gene here is read or not. So for each one of these 20,000 players, imagine about 100 of their relatives, their friends, their grandmothers, their aunties, their Facebook friends, who have an influence on what they do. And in the end, how the cell develops and how the cell ticks is the combination of these 22,000 players and millions of areas throughout this genome that have an influence on what they do. And that's truly daunting. And that's one reason why, even though we have the whole genome of thousands of humans, we haven't understood why you get cancer, why you age, why some people die sooner, why some people are unhappy, or why some kids develop non-typically. And so it turns out even these enhancers can be transcribed into RNA that then swims around in the cytoplasm and instigates. And this is an example of the beads on a string, and there is a gene there, and you see these purple and pink things. Those are enhancers that have an influence on whether that piece of DNA is expressed. And this is different in different tissue types. And I apologize for the overexposure. To your right are mouse embryos that show the expression of these different genes under different enhancers. So in terms of RNA, almost every week people describe a new type of RNA that hadn't been appreciated before. And it turns out that really this is not mostly junk. A lot of this stuff is transcribed into RNA, which never leaves the cell. But this RNA can fold up and make little tools, and these tools swim around the cell, and they influence gene expression. So when we talk about how DNA makes you, you have to keep in mind that there is like a syntax of gene regulation. I've already mentioned that the way DNA is packaged contains information. Those packaging spools can themselves be modified chemically. And that modification changes. If you're diabetic, they are modified differently than if you were healthy. If you're exposed to carcinogens in the environment, it changes how these spools are modified. DNA itself can directly be chemically changed by a methylation. That's the classical epigenetics. I mentioned the non-coding RNA, and to make matters more complicated, there are proteins that never leave the cells. They swim around inside this vast ocean of every cell, and they interact with these little pieces of RNA. And so in the end, how the cell ticks is not just the 22,000 genes. It's all these moving parts together that allow you to develop into who you are. And the next time somebody asks you, now somebody talks about epigenetics, probably the best trick to remember epigenetics is, you all know bees. Most bees you see here are worker bees. They're females. They were laid as an egg by a queen. These female bees can decide that they need a new queen. When they decide that, nobody knows how they decide that, they make a different food. They synthesize the food in their body. That food has an enzyme that changes the annotation on the genome of the egg. And the exact same egg that would give you a worker, usually, if it's exposed to royal jelly, will give you a queen. That is 2 to 3 times bigger than the workers and will start laying eggs. So the worker bee to queen bee transformation out of the same egg with the same genome is based on messing with that annotation of the DNA known as histone modification. It's not the DNA itself, it's the spooling of the DNA that has been changed. And that's my favorite example for explaining epigenetics. So now we jump back to transmission of DNA, and I'd like to convince you that it's almost impossible to divorce culture from genomes. And here's an example. So fairly recently, a group of people at Stanford looked at Y chromosomes around the entire world. Remember, that's the small chromosome that never recombines. It just is inherited by all the males in the group, and those of you with kids will transmit it to your boys. If you look at the diversity of these in different colors, the deepest branches are within Africa, yet another proof that we're all African. And then the ones after humans left Africa, there's a couple of little mutations that light them up in different colors. So what you can do is you can compare mitochondrial DNA to your right to Y chromosome DNA, and the higher that bubble, the more gene-rich, the more diversity there is. And you will notice that between 70 and 50,000 years ago, both the Y chromosome and the mitochondrial DNA had many, many more copies. That's known as the out-of-Africa bottleneck. That's when modern humans really went to town and started colonizing the entire world. And it never stopped for the mitochondrial DNA. There's more and more and more diversity. Now watch to your left. There is this dip in how much diversity there is in the Y chromosome. This is as if there were thousands of last names, and suddenly everybody is called Smith. Now, this timing is about 5,000 years ago, that smack in the middle of the last 10,000 years since we invented agriculture. What this shows you is that some guys got much more lucky than most other guys. Some males are totally overrepresented and pushed out other males from the gene pool. And this is cultural. This only was possible, this was never possible with hunter-gatherers. This is an event of the Neolithic. Farming, you can have ownership, you can become powerful, you can hire armies and priests, and then you tell everybody else that they can maybe have one wife, but you have 300. And what? You can do it, you can see it in our genome. So you're looking at a pattern in the human genome around the world that is caused by culture, inventing agriculture and becoming very non-egalitarian, where some males like Genghis Khan and kings of Morocco can have hundreds to thousands of mating opportunities. It doesn't end there. If you take this pattern here, and this is from left to right, it starts in Addis Ababa, Ethiopia, and it goes the furthest away from Africa you can to Patagonia, which was last colonized by Native Americans. And the higher these violin plots are, the more inbreeding you have. The bigger the chunks of DNA that haven't had time to be shuffled. In the red box is an area where there is more inbreeding than you expect. The genome shows a pattern that huge chunks of DNA that you would expect to have been shuffled haven't been shuffled. Why not? It turns out these are societies where it's regular or even expected that uncles will marry their nieces or that first cousin will marry. For cultural reasons, for economical reasons, because uncle-niece mating is pretty nice to keep the family ownership together. Same for first cousin mating. This is cultural behavior that leaves a print on our genome. So next time you talk about genetics to someone, don't forget that genetics doesn't happen in a vacuum. Genetics happens in a social, cultural society. So if you're interested in things that differentiate us from a great ape, there's no end to it. You pick up any dictionary and you start with the letter A, astronavigation, beekeeping, cycling, x-rays, yoga, ziplining, thousands and thousands of things that we do and no ape, at least apes not trained by humans for thousands of hours, have ever done. This has something to do with the fact that even though chimpanzees have culture, they use tools, they take years to learn how to use these tools, different groups of chimpanzees have slightly different culture. What they seem to lack is what we call ratcheting culture, where in human society somebody has an idea, oh, instead of using the big tear-shaped hand axe, I'm actually going to use the little pieces of obsidian that flaked off and I'm going to mount them on something that I can put in a bow. Now I have a bow and an arrow. And somebody copies that, and then somebody improves it, and then you make a crossbow, and so forth. So humans have ratcheting culture. We build upon ideas of others because we have language. We can exchange ideas. We innovate and we copy. Copying is just as important as innovating. Most of us are not innovators, but we're all really good at copying, which is important. That's how we keep good ideas in the group. So a couple of field primatologists who spent many, many years studying non-human primates, looking at their communication, at their tool use, at how they can, whether they empathize with others, how much they cooperate, they have argued that, yes, apes have culture, but they kind of act as if they wouldn't know that they have it. And they call this the Jourdain hypothesis. It's named after a character in a play by Molière, a very comical French theater writer, where this guy wants to learn to talk how a gentleman, like a gentleman, and then he comments that, oh, I didn't know that I was speaking prose. So it's called the Jourdain hypothesis, and it's quite convincing that there is no evidence that apes, unlike humans, most human cultures are self-aware about their cultures. Most cultures, especially the fun ones, make fun about their own cultures. They are aware that they do things differently than others. And that's something that there is no evidence for in non-humans. So there's something going on about self-awareness, which leads us, this is exploring Darwin. In Darwin's view, the fact that humans blush is one of the most peculiar of all human emotions. Think of it. You cannot blush when you're alone. You have the biggest organ in your body, your skin, that uncontrollably changes color when you start thinking about how others view you. So this is known as theory of mind. You use your mind to have a theory of what others might be thinking about you. You try to plug into other people's minds. We love doing that. Many of us are obsessed with doing that. That's why we have Facebook and all these things. And it turns out that blushing is an uncontrollable reaction that tells an audience that you're self-aware that you might have done something embarrassing, wrong, or, and you can't control it. Obviously, if you have less melanin, it's more easy to detect it, which is an interesting question on why some people should have lost melanin. What's interesting is you apparently do it in the dark. There's a group of people who found a lady who was famous for blushing very easily, but they controlled, if she was alone, she couldn't blush. But if she sat in a dark room with two people and told a story about herself where she was embarrassed, they could measure with a heat sensor that she was blushing. All humans blush. It's just more easily detected in people who lack melanin. Very important, language. All animals communicate. Language is a very special form of communication where you can create meaning. You can create an infinity of meanings. I can tell you a story about fictional characters that have never existed, and you will start having a theory of mind of these people like Sherlock Holmes, or pick your favorite character from a movie scene. You will think about the mind of someone that never existed that I made up or somebody else made up. That's how powerful language is. I can take you back and travel through time. I can tell you about mistakes I made. I can tell you about mistakes my grandfather made and he told me. And then you can take this on board if you decide to do so. We don't know of any other animal that has this option. It weaves together populations of minds. Ideas can survive. I might be really bad with my hands, but I convey to you something that my uncle had built, and you might be able to then build that. So the idea didn't die out, even though I wasn't capable of building that machine, because when I tell it to you, it inspires you, and you go back and try to build a machine that only existed as an idea, but through language, I can keep it alive. What is really important is what language, in combination with names, allows is reputation. You know that if you do something that I observe, because I have language, and I might know your name, I can share this with thousands of people. And we all know this, and this has a very strong influence on what we do and what we don't. Just watch some of the comments that people can make online when they can be anonymous. You can instantly see what happens when reputation is lifted out of the system. This is why things like Airbnbs and PayPal, they all have reputation grading, because reputation is an incredibly powerful form of social control. Then, to get back to my funny accent, human language is this incredibly honest signaling. I can say one syllable out in the surf in the lineup. I say, hi. And they go, oh, where are you from? One syllable. I've been here 23 years. I cannot pretend to be from here. There's a famous law in, in the FBI and the spy world that if you haven't lived in a society by age 12 or 13, you can never pretend to be from that society. So some people have commented how odd that language is a human-specific communication system that evolves to become difficult to understand. So part of what carries language is this huge, big brain. Humans have a brain that is 3 times bigger for our body size than chimpanzees. And a lot of that happens in the womb and then after birth. What drove this big brain? We still don't know. But we have fossil evidence that over the last 2 million years, combined with the technology, first a lot of stone technology, which is the only thing that fossilizes. We can never find ropes and traps and nets and baskets because they rot, but maybe they were really important. Over the last 2,000 years, human ancestral skeletons we find throughout Africa first, and then up into Georgia, and then all the way to Indonesia. There is more and more room in the head for a brain. We don't know what these brains looked inside. Very recently, a group of Japanese scientists managed to convince these are chimpanzees that are fully habituated. They gained trust to a point where the human scientists could actually do an in utero ultrasound throughout the entire pregnancy of this chimpanzee. And what they found is that, unlike in humans, chimpanzee brain growth plateaus in the middle of gestation. We power straight through. Not only that, after we are born, we continue developing our brain at a fetal rate, a rate only known in the fetuses of other mammals. So humans give birth when it's pretty precarious already because of bipedality. So that was a very big cost, making that big brain, because many mothers have died in childbirth and so have their children. But apparently that was worth it. The brain continued getting bigger and bigger and bigger. But after birth, that brain doesn't slow down. It continues growing. Now it's being sung to, it's being held, it's being spoken to, it's being enculturated as it forms its connection and starts working. That's the incredible cost. 3.5 million years ago, Australopithecines that were clearly bipedal, as we can judge from their skeletons, started having issues with the head, but nothing like we face now. The other thing that changed is the schedule. After you're born, how long are you a child? Do you have teenagers? And it looks like, based on data from an old Swiss-German primatologist who spent 25 years in Johns Hopkins and looked at thousands of great apes, humans seem to be unique in having this period of childhood where the body stops growing very quickly, but the child is developing mentally and learning language and learning what people do and what people don't do. You don't see that in the great apes. They seem to be more on a steady state growth. Is this schedule of development adapted to cultural opportunities or nutritional opportunities? Is it facilitated by the fact that in most great apes, it's just mom and the little one? In many human hunter-gatherers, it's the kid, the mom, the dad, the auntie, the grandmother, and so forth? And is it promoted by allomothering, which is people other than mom and dad looking after the young ones? So this growth schedule allows us to develop this incredible mind. And really, our minds are first and foremost a copying machine. The target of that copying is mostly language. Mind you, human children learn multiple languages with zero instruction. All they need is exposure. It turns out if there is little exposure, they don't learn as well. In our DNA must be something that tells our brain to develop for language, because those unfortunate kids who are not exposed to language have a brain that is insulted by the absence of language. So how did this ever happen? How did we integrate the need to be exposed to language into the genes that tell our neurons how to behave? It's a question that many people are trying to answer. Obviously, if you have good copiers, you're much better at keeping innovations and developing slowly might give you this beautiful opportunity to take on board what everybody does where you grow up. But it's not just in early age that humans differ. What is strikingly different, this pyramid of age here shows you survival of individuals in chimpanzees and in human hunter-gatherers. The purple bars up there are post-reproductive individuals, mostly women. For a long time, people thought that human menopause is some kind of disease that needs to be treated. It's now becoming very clear that human menopause is a species-specific adaptation to keep grandmothers alive, because their contribution to the next generation is so precious. And they would be risking their lives by trying to continue to reproduce. Now, recently, with a couple of colleagues, we were interested in a gene that we study in the context of immunity, and this gene was discovered by other people to be involved in Alzheimer's. Well, it turns out that we seem to have found a mechanism by which an old Darwinian dogma might not hold in our species. The idea is that it might be worth keeping older people around because they increase the survival of their young kin. We found a whole series of genes that hadn't really changed much between chicken and chimpanzees, but they change in humans, and in humans the change protects you against dementia or Alzheimer's disease. This one particular gene comes in two versions. If you have too much of one version, you have a higher risk for Alzheimer's disease. And it's only been changed in humans. Chimpanzees don't have that. And so we argued that this is an example where natural selection acts on the group of people that is supposed not to have any selection left at all, namely post-reproductive grandmothers or grandfathers. And it works by them benefiting their younger kin. So classically in evolutionary biology, you can experimentally show that natural selection almost disappears after you hit the average age of reproduction. There is no more selection. It's really important early on. You have to survive. You have to have kids. Remember, natural selection doesn't push you to become healthy and happy. It pushes you to out-reproduce your neighbors. However, this is an interesting mechanism where in a species with language and the transmission of ideas, you can continue selection that benefit the old, but through the survival of their young. Interestingly, how you rear children might also have an effect on that. A recent work on human dads has shown that the levels of testosterone in men drops the more time they spend with their young children. This is a very interesting effect where, depending on who cares for the baby, males that usually between 20 and 30 are at their peak aggressiveness and most likely to be involved in wars, would get a little break from testosterone poisoning based on the cultural convention whether dad looks after children or not. Tellingly, in many hunter-gatherer societies, the fathers spend up to 50% of the time holding the young baby. But as soon as you have pastoralism, farming in the last 10,000 years, the dads are not around for the first year, and you see no drop in testosterone. So this would be an effect where the baby, via culture, namely who holds baby, has an effect on the biology of the father. So we now have complete genomes of humans and several chimpanzees and other great apes, and this is a grand summary from a review that a couple of colleagues and I wrote a couple of years ago, showing that almost every possible change you can imagine exists in the genome. Some genes, we have more copies or less copies than chimpanzees. There are completely new genes, a piece of DNA that meant nothing, that now becomes a gene in humans. Pseudogenes are genes that are still working in a chimpanzee, and in our case, that same gene has a stop codon. You can recognize it, it's there, but it's not doing anything anymore. Positive selection is a term that means the DNA has changed more than you expect by chance. And one of the Hall of Famers of positive selection is called Human Accelerated Region 1. This is a little snippet of DNA that doesn't code for a protein, it codes for one of these RNAs. It's 118 letters long, nucleotides long, and it has 18 changes between humans and chimpanzees and chickens have the same sequence. So something that evolution has held to stay exactly like that, and then something in our lineage happened in the last 5 million years. We changed this like crazy. It's involved in the cortical layering of the brain, so it's almost impossible to study. You need human embryos to study this. You can put this in mice and see where it's expressed, but mice don't make a human brain. So at this point, it's almost impossible to do what this does. But it's a very, very fascinating example of certain chunks of our DNA have been tweaked like crazy in relatively recent time. This is what you can do with some. This is another region, human accelerated region 2. You can put this into a mouse embryo, and using reporter genes that express an enzyme that can cause a blue color, you can tell where this gene is expressed. And this is interesting because it's expressed in the limbs. But again, these are the limbs of a mouse, not of a human. And you can't do the experiments in non-human primates because it's ethically not allowed. Many of these genes that have been tweaked recently seem to be involved with growth rate of our brain. How much do cells divide? How much do they migrate? How much do they contribute to this bubbling brain that we have? More recently, people who are real pros at studying the entire genome have started looking at chunks of DNA that have copied themselves like crazy. Here an example where chimpanzees and bonobos have all these copies of that same DNA sequence across their entire genome, and we just have 3 copies. So this is an example where this DNA doesn't even exist in us because it just copied itself, but only in the great apes. Well, the reverse also exists. This is an example where humans have multiple copies of something, and the chimps don't. And this is really interesting because this evolved in the last million years. And if you put these extra genes in a mouse and look at its brain, the brain has extra synaptic buttons, and the way that the neurons form connections is increased when they have the human copy. So this is almost one of the smoking guns of one of the things that had to be tweaked to get a different brain. You can then look at neuronal development in these mice, and in fact, even in the mice, you can tell if you put the human version of this piece of DNA in, their neurons behave differently and behave more human-like in the brain. This is to show you the nightmare it is to study a genome that has cassettes of similar sequence in them. But if you can navigate this, you can actually find out that some of these cassettes have copied themselves, and they have set up uniquely in humans, so the last 4 lines, each one is a haplotype. The lower 4 lines are haplotypes. In humans, you see that red area, the 550 kb critical region, because the same DNA has copied itself on both sides of it. This now tends to fall out or to copy itself an extra time. So this is like an area in your genome that has become unstable because 2 similar sequences are found on both sides, and it can copy or fall out. Well, this gene is called BOLA2, and it turns out there's 1 copy in the great apes, there's 1 copy in Neanderthals and Denisovans, and all modern humans from around the world have 8 or more copies. Why is this important? If you compare populations, you find certain individuals that have fewer copies and certain individuals that have more copies. Anybody who has less than 8 or more than 8 is on the autism spectrum. So this is an example of a recent tweak that puts you on the knife's edge of having a modern human mind. But if you're unlucky and your genome hops away from that through mutation because it's not stable, you crash and you find yourself on the autism spectrum disorder group, which is a very interesting topic because many of these genes that have uniquely changed in modern humans seem to be involved with brain development and deviations. I mentioned how slowly we wake up. This is a study by Chet Sherwood and colleagues showing myelinization. When neurons make connections, once these connections are established, you wrap them with myelin to consolidate the connections. This is done by age 10 in a chimpanzee. Those of you who are between 20 and 30 are still in the process of finishing your myelinization. So humans continue maturing their brain into their 3rd decade. A group of scientists in Shanghai with Philip Kaitovich has been able to access dozens of brain samples post-mortem of humans, chimpanzees, and macaques and look at gene expression. So these are not induced stem cells, or neurons from stem cells. They are actually brains. And they find that roughly the same genes are expressed, but they are expressed later in humans. So this is known as neoteny or heterochrony, which means you get a different animal by using the same genes, but the timing of when you express them is later. The solution to get back to that crowd of 20,000 people standing in a square and having to know what everybody is doing to everybody else, what people now do who study brain expression in the brain, they look at networks of hundreds of genes, and they ask, are there networks of genes when one gene is expressed in humans? There is 30 other genes that are all also expressed at the same time. But in chimpanzees, that same gene is expressed, and there's just 2 other genes that are expressed. And so people are now slowly starting to finger entire gene networks. And these gene networks show up in very famous human psychiatric disorders. The most common is anxiety disorder, which hits about 1/3 of all humans during their lifetime. to much rarer things like schizophrenia and so on. But there's a lot of interest in the genetics of psychiatry in genes that involve neural function and brain function. People are now using these gene network approaches to try to disentangle entire webs of genes. Remember, 22,000 genes interacting with hundreds of things each. It turns out most traits, these are different examples, ADHD, autism spectrum disease, schizophrenia, and so forth, mood disorders, ADD. What these things show you is that something like schizophrenia is not caused by one gene. There is not a schizophrenia gene. There are hundreds of genes that, if tweaked slightly, might put you in the unfortunate position of having schizophrenia. Just like there are hundreds of genes that tweaked the other way might put you in a position of having a very unusual, highly functioning mind and being a very nice person at the same time. But the numbers that are being tweaked are almost beyond comprehension. I'll jump over FOXP2, I'll jump over this too because it's a bit too much. That's the same exercise of trying to study how faces of humans and chimpanzees and great apes have differed based on enhancers, these pieces of DNA far away in the genome. And I'll end with a reminder that DNA and proteins is not everything. This is an example of a mutation that happened in humans about 2 million years ago. This is something I personally have worked on with my colleague Ajit Varki, and we've published quite a lot on this. It's a tiny change in DNA, namely this enzyme lost just 400 letters. 92 of these letters were in an exon. It lost its function. So now humans, unlike all other mammals, cannot change one sugar into the other sugar. Tiny DNA difference. It changes the nature of one sugar, but look what it does. If you make yourself the size of an influenza virus or a malaria parasite and you land on a red blood cell, that's what you would see. The cell membrane, and outside the cell membrane, a jungle of sugars. And the last little sugar in red is called Nu5AC if you're human. If you're a chimpanzee or a cat or a dog or a pig, it's two different sugars, which means that the surface of every one of your cells has a different flavor. And that flavor is really important because it determines which disease can get you. So what we have surmised is that about 2 million years ago, humans managed to escape malaria in Africa, because 2 million years ago, the only humans in existence were in Africa. This gave us a nice break from malaria. Unfortunately, with farming, malaria jumped back into humans with a vengeance, and this is the malignant malaria that can kill you overnight. This gels pretty well with the species radiation of the mosquitoes that lived in the places where there was farming. So that's an example where a tiny change in the DNA can change the surface of every one of your cells. So you are more than just your DNA. So in summary, I want to convince you that, hopefully convince you that individual genomes are unique genetic mosaics. You each exist as that particular genome for one generation. And then you scramble it again for the next generation. Each piece of DNA has a distinct history. Some pieces of DNA in our genome come from Neanderthals, now extinct relatives, but that's the tiny minority, and they are usually not where there is action. Complete genome sequence for many apes allow us to reconstruct past population histories and find changes that define human species, but it's still a working agenda. We're very far from clear answers. What is clear is that we are enriched in changes that affect brain development, including genes involved in uniquely human diseases. Fossil DNA data and experiments in model animals and cell cultures, as some of you have heard earlier this morning, allow you to test for biological effects. But the experimental approaches are really, really limited because we can't do things in non-human primates. You can't make a transgenic ape. There are some efforts afield to make transgenic marmosets, which are tiny New World primates, but they have very different brains than humans. So with that, I thank you very much. And